So I want to welcome everybody to this new guides workshop on biostatistics. And this will be presented by Dr. Denise Roe. So let me tell you a little bit about Denise while we're waiting for people to show up. And Denise is a professor in public health here at the University of Arizona. She's also the Shared Resources Director for Biostatistics and Bioinformatics at the Cancer Center. She's got a team of biostatisticians that she works with and oversees. She received her BA in biology from UCLA, and then she went over to the dark side and got a master's in biometrics. Um, then she went on to get a doctor in public health at UCLA and came to the University of Arizona in 1988, where she became a faculty member and has been actively involved in multiple grant projects. She's reviewed grants for NIH. She's been instrumental in clinical trial design for a couple of big trials. And it's really a pleasure to have her here to talk with us today. She likes questions and be happy to answer those. And she'd like to work with you. She's worked with Nicole and Pavel, and she'd like to extend her base. Take it over, Denise.
Okay, well, thank you, Ron. Let me share my screen and then put it on screen slideshow. Okay. So what I will do is I will hide the video, but if you have questions in the chat, please either just interrupt me or put them in the chat, and I trust that Ron and Maria will direct them to my attention. So as Ron said, I am the director of the Biostatistics and Bioinformatics Shared Resource in the Cancer Center. I'm involved in both of the current NACP projects that are ongoing. And I like this slide as a title slide. I'm indebted to Dr. Aaron Scott for this slide. And I think it is a very appropriate slide for the biostatistician and our role in grant development. Because as you can see, even though the road is smooth, it is not paved. It has twists and turns, and you can't see around the horizon. But I think this is a very appropriate metaphor for what we do in research design and grant development. So today, what I want to talk about, and yes, I have worked with Nicole, so she's very familiar with this. I want to demystify the process of consulting with the statistician. I want to talk about what you as a researcher bring to the collaboration, because this is really important. With respect to grants, I'm going to talk about the importance of the specific aims page. I'll talk about what I, as the biostatistician, bring to the collaboration, the grant sections that I can help prepare, and then statistical landmines to avoid in grant development. At the end, I'll talk about a really nice example where we turned an R21 grant into an R01 grant, so you can see the process in terms of how these statistical concepts evolve through a research program. And then I hope there's time, well, there should be plenty of time if you're interested for us to discuss statistical design considerations in the context of the proposals that you're developing. So then what do you bring to the collaboration? Well, I assume that you have a grant idea, you have a hypothesis. It's helpful if you've thought about a particular RFA or a particular grant mechanism that you want to respond to. But basically, what I need you to bring to me is, what's your hypothesis? So if the study is successful, what do you want to show? and then an outline of your specific aims. Usually most grants have 2 to 3 specific aims. So then what do you propose to study to address your hypotheses? You need to tell me why this is important. All grants start with a significance section. So what is already known? What's the context of that you're working in? Are the previous studies scientifically rigorous? And this is really important in terms of you setting the stage for why what you're trying to do is important. And then, can you bring me an example of anticipated results based on the current literature? So if you can bring me a paper that measures the outcome you're interested in, even if it's in a different patient population or a different patient group, or pilot study results, that helps in the design of your study. Now, innovation is very important in any grant submission. So what's novel about your study? And you have to be very careful when you write this section. That means you have to have done a very thorough literature review. But then it's important for you to spell out for your reviewers the innovative aspects. So in other words, if you're assessing a particular intervention in a new population, or if you've modified an intervention that's used in another population for your population, what's innovative about what you're trying to do? Then we'll talk through your approach. I always tell investigators that come to me with a grant idea, I will ask you a lot of questions. But who's your target population? Who are you including, and who do you feel you specifically should not be studying? What are the primary and secondary outcomes of interest? The primary outcome is what we're going to use to determine the sample size of your study, but secondary outcomes are the other outcomes that you're interested in. And then Dr. Jimenez is part of a group that's outstanding at using figures to summarize the approach. So if you can develop a nice figure that shows the sequence of what you're trying to do, that can be really helpful. Now, how will you measure your primary outcome? Okay, for example, in the study I'm going to talk to you about later, we were interested in measuring hemoglobin A1C. Statisticians prefer what's called hard endpoints. These are things that are quantitative and easy to measure. So things like BMI, weight, waist circumference in clinical trials. We often use survival or progression-free survival as the endpoint. If you're going to use behavioral endpoints, like many of the studies that I use and I'm involved with in cancer patients, use quality of life outcomes as a secondary outcome. You want to make sure you're using a validated questionnaire in your study population. A mistake that junior investigators often make is their first specific aim is to develop a questionnaire to measure something in their population of interest. Developing questionnaires is much harder than you think it is. And in fact, if you feel you need to develop a questionnaire, that should be another grant submission, not part of your primary grant submission. So it's better for you to use validated questionnaires that have already been used. And within the Cancer Center, the Behavioral Measurements and Intervention Shared Resource that's headed by Dr. Jem B is outstanding with experience at validated questionnaires to measure behavioral endpoints, things like diet, physical activity, sleep. Those are the behavioral type endpoints that we're talking about. And then what are your secondary outcomes of interest and how will they be measured? you should really limit the number of secondary outcomes. And in particular, when you get the grant funded and you do the analysis, you have to be really careful about overemphasizing the results of secondary outcomes, because typically the study was not powered to detect them. So then what are your specific aims? Even if you just bring me a draft, it should include abbreviated background, tell me the significance of what you're doing. Your hypothesis should be very clearly stated, your specific aims, and the potential impact. You should draft that first. I've had some junior investigators say, well, I started writing the methods and then I'll go back and write the specific aims. I've worked with several very senior investigators that will take a month or two to draft the specific aims page, really hone that in, and then start to write the rest of the grant. Now, I'm going to emphasize a couple common mistakes in specifying specific aims. Often, junior investigators have process aims, not hypotheses. So an aim is not assemble a cohort. An aim is related to a scientific hypothesis. Or they combine specific aims. So for example, to test the efficacy of the intervention on changes in body mass index and its feasibility and acceptability to the study population. As you'll see when I get to the example at the end, we clearly separated efficacy from feasibility and acceptability. Now, let me pause here for a minute and see if there are any questions, because now I'm going to talk about what the statistician does. Pavel, you're an expert at making these really nice figures.
Thank you, Denise.
Yeah,
definitely they help. Yeah.
Diana, I don't know you as well. I work with Pavel and Nicole on a regular basis, so I look forward to working with you also.
Thanks so much. I really appreciate the opportunity, and it's very nice to meet you as well. And maybe I should introduce myself a little bit so you know who I am. So I don't want to take too much time, but I'm Diana. I am from environmental engineering actually, but I work with Jenny Ingram. Maybe you are familiar.
I know. Yes, I do know Dr. Ingram.
So she's my main mentor, and we work together in a project that we look in water quality and how water quality is related to cancer in Native American communities.
Okay. Well, that sounds fascinating.
Thank you.
Okay, so what do I as a statistician bring to the collaboration? Well, I take your research goal and I translate it into a statistically testable hypothesis. So the outcome that you propose determines the method used to estimate the required sample size. Now, often we have a fixed sample size versus a sample size that we do have the capability to expand as needed. It's always better to work with me early, and Nicole and Pavel know this well, with respect to the study design, and usually you need the required sample size to be able to complete the budget. Maria and I are working with a NACP project now where the design has evolved as the study is ongoing, but we have a fixed budget. So we're having to redesign the study to meet the constraints of the fixed budget. So what's my role? Well, I determine the appropriate method of statistical analysis, which is what I do to determine the sample size for a given power and difference to be detected. I develop the statistical considerations and sample size justification sections. I always write them in that order because I have to talk about how we're going to do the analysis first, and then I can write about the sample size justification. And usually we consider the use of sex and age as biologic variables. Just to remind you, what do we mean by sample size and power? Well, anytime we design a study or analyze a study, the alpha level is what we set so that we know that roughly 5% of the time, we have the risk of concluding there's an effect when in reality there is no effect. The power is the probability that we conclude that there is an effect when in fact it really is there. So when in fact there is a difference between, say, 2 treatments or 2 interventions, we want high probability of determining that that's true statistically. It depends on the statistical analysis approach. We usually select the sample size so that the power is typically 80% or 90%. We need to adjust for planned interim analyses. And then we also need to adjust for multiple comparisons. So this is typically why we only have a single primary endpoint. So as an example, current approaches lead to a smoking cessation rate of 15%. We want to improve it to 30%. We want to do a randomized clinical trial of a new intervention technique versus just standard of care. And we wanted to have at least 80% statistical power. Okay. Well, you can see that if we only have a sample size of 100, we would have 72%. 120 would be 79%. We would need at least 140 subjects to have greater than 80% statistical power, and 180 subjects leads to greater than 90% statistical power. Okay. So what if we can only accrue 100 participants? Well, going back, you see that, well, we won't have 80% power. So instead of having to have an improvement from 15% to 30%, to have 80% power, we'd need to have the improvement be at least 31.5%. So we would round that up to 32%. So in the sample size and power justification, it's important that we define the assumptions that we're using. We provide pilot data or results from the literature to justify the assumptions. And then I typically provide enough detail so that the biostatistical reviewer can match your calculations. Almost all R01 grants have a separate biostatistical reviewer. So I tried to give them enough detail, even to the point of telling them the program that I use to generate the results so that they can match them. The proposed statistical analysis plan is tied to the scientific objectives. I clearly state the primary outcome and all secondary outcomes. I recently worked with a prostate cancer program project grant with Dr. Ann Kress and her collaborators. And that was something that I worked very hard in the statistical considerations to do, to take their projects and for each aim, specify the primary outcome and the secondary outcomes. I summarized the proposed analyses in their study. Because it was prostate cancer, the sex was not a biologic variable, but we did need to talk about age. And then discuss potential biases, the potential for missing data, or other limitations. So what are the statistical landmines to avoid? These are things that if you have them, your grant will almost never be funded. If you don't have a sample size justification, usually you will not be able to get a grant funded. And if you say it's just based on feasibility, which means budget, you need to clearly justify that you'll still be able to identify an effect that's meaningful from a clinical or public health perspective. I've seen people do things, sample size justification based on previous significant results. That's not an appropriate sample size justification. A generic statistical analysis plan, I've actually seen people just basically take the statistical methods from a paper and embed it in their grant application. And, that just doesn't work. And a naive statistical analysis plan. If you have clustered observations, and I'll show you an example where we had to include the fact that we were going to have cohorts of individuals, we need to account for that in the analysis. Okay. So again, let me pause for a minute because this is a good point to take questions.
I have a question with the power, because like in our microbiome studies, frequently we struggle to know all the effects, right? And there's like multiple effects, and when we're doing that together with some extra omics, like those factors are many times not known, or the effect size it's not known. How you kind of like deal with this type of data or study designs to kind of handle that?
Well, Pavel, we've spent a lot of time talking about that. I mean, usually what you've done is, I think, very appropriate. You look at the effects that are at least a doubling or a tripling. Usually, and we can build this in sample size estimation, we can use a false discovery rate approach to adjust for the fact that we may be looking at 1,000 different outcomes. So that's another way that we can get around it. But you're right, especially this issue of multi-omics integration, that actually, as you know, is an area of very robust statistical research in terms of both how do we do it, if we have genomics, proteomics, in our case, microbiome, how do we integrate those? But then if we're designing the study, how do we show that we'll have an adequate sample size? So that's an open question statistically.
That's what I remember from our previous conversation.
Yeah, now I wish there was, okay, well, let's look at
Right.
this paper, but no, it's not that simple.
Hi, Denise. This is Melinda.
Hi.
First, I love how you're breaking everything down. When you don't do these things very often, it's a great reminder. So my question is, I am designing a community-based participatory research collaborative project with a very small community, and the total population is really small. So I'm looking at people with type 2 diabetes within this community, and I'm struggling with this whole determining sample size, especially if it's just going to be a small pilot project. So I don't know, I would love your insight on maybe past projects and how that was done.
Okay, well, that is a very good question. So let me come back to that after I show you the first part of the example that I'm providing. Okay, and then we can talk about it a little bit more, if that's okay.
Yeah, that's great. Thank you.
Okay. All right, so this was a grant I did several years ago with Dr. Melanie Hingle in the Department of Nutrition and the College of Public Health. It was a R21 grant that we got funded. And EPIC stands for Encourage, Practice, and Inspire Change. This was a YMCA-based type 2 diabetes prevention program for overweight and obese 9 to 12-year-old children and their parents and caregivers. So in this R21, we had enough budget. An R21 is a grant that's high risk, high reward. So that you don't need to have, you'll show how we justified it, but you don't need to have as rigorous of a sample size justification as you need for an R01 grant. So based on our budget, we could do an initial sample size of 60 parents with their children, because we knew that we would have about 20% attrition, which means we wouldn't be able to get their final measurements. We were going to do baseline measurements and then after the intervention. And it was based on previous results in a YMCA-based childhood obesity program. So we had two formats: a face-to-face delivery led by a YMCA lifestyle choice coaches, and a hybrid delivery with alternating face-to-face and mobile device delivered content. The primary analysis was based on a paired t-test. So we were doing pre-post for change in BMI z-score. Since these are kids, we can't just use their BMI. We have to use their z-score, which comes from tables that are constructed by the Centers for Disease Control and Prevention that takes into account their chronological age. And our primary analysis was combined across the two formats. We were going to do exploratory analyses to compare differences between the formats. So at the end of the study, we had 48 children, and their average age was around 11, 45% female, 54% Hispanic, 87% obese, and their parents. 36 families completed the post-intervention measurements, and program attendance averaged 61% across the intervention. What we saw were significant changes in the child BMI z-score. So we saw a significant decrease at 12 weeks. We saw a significant improvement in the family, food, and physical activity environment. And the participants and lifestyle coaches highly rated the program content and engagement strategies. So this was our pilot study. Now, let me just spend a minute talking about the advantages of a pre-post design. It can allow assessment of the feasibility and acceptability of an intervention. It can provide a preliminary assessment of whether the intervention leads to a significant change, which can provide estimates of the effect for the design of a future randomized trial. What it can't do is it can't rule out interfering events outside of the intervention. So if, for example, while our intervention was going, we couldn't account for the fact that there might be multiple news articles about the detrimental effects of obesity in children. Just the fact of assessment itself may affect the outcomes. I mean, if people know that they're measuring their BMI, their weight, their waist circumference at baseline, it may affect what they do during the study, irrespective of the intervention. And we can't rule out what's called regression to the mean, which means the more extreme a score is, the more likely it is to be closer to the mean at a later measurement. So let me pause here and go back to the question from Melinda about your pilot study, and let's talk about what you're trying to do.
Yeah, so this is going to be a traditional pre-post test as well, pre-test, post-test. I'm looking at knowledge gained as well as, so it's a mixed methods. It's knowledge gained as well as cancer screening intentions and health behaviors.
Okay, so are you restricting attention to patients with participants with type 2 diabetes?
We are.
Okay, so and you said you were in a particular tribe.
Yes, and so the total, what was it? It would be 30% of individuals about there 18 years and older have type 2 diabetes within the community. which sounds like you'd have a lot, but in this particular community, which I'm keeping them anonymous just out of respect, it's not that much, that many people, and those are individuals. We're looking at just individuals who still live within the borders of the reservation. So
Okay.
I can't give you a specific number, but the total population of type 2 diabetic people living with type 2 diabetes is pretty small who live within the community.
So it sounds like what you're basically doing, since it's a mixed method study, is you're doing a feasibility study. Have you developed the intervention materials?
They are, yeah, we developed them. Yes, they're developed. Sorry.
Okay. Okay. So you're using existing materials.
Mhm.
And then how are you going to track their changes in knowledge, attitudes, and behaviors?
It will be using a survey, so it will be administered before they complete the health education intervention, and then afterwards, and then there will be a 3-month and 6-month follow-up. And the follow-up will be just qualitative focus groups.
Okay. Now, this is the hard part. And basically what I'm modeling is what I do in statistical collaboration. When you measure their knowledge, attitudes, and behaviors, are there validated questionnaires in any Native American populations?
Not that we found. So we've adapted this next step, we're going to adapt an existing survey looking at cancer screening intentions, but it was created, I think, in Britain.
Oh, I'm surprised. Now, there's other cancer screening
Mhm.
intentions surveys.
Yeah, I'm still open to it. It's just the one I found seemed to fit what we're looking at.
Okay, but screening for cancer. Okay, what's the primary focus of your intervention? Is to get them to screen for cancer or to change their behavior related to type 2 diabetes risk factors?
So the overarching goal is to reduce cancer risk by improving health behaviors around diabetes management and cancer screening.
Okay, okay. All right, so I would imagine there's validated questionnaires, but they may be in Anglos on diabetes management. So, and because diabetes is so prevalent in Native American populations, yeah, who are you working with?
So this one, I'll be working with it's right now, I haven't been awarded it yet. So we would be working with I would be working with Nicolette Tofelshon and Dirk and then the folks out at the health department in the
Yeah, no,
community.
I know Dr. Teufelschoen. She and I have worked together a lot when she was at the U of A. And then Dirk, I'm actually working with him on their grant now. So I know both of them. So boy, I'd be surprised that there aren't surveys in Native American populations about diabetes management.
Right now,
So
oh, sorry.
go ahead.
Right now, the screening, the survey that we developed, we did use, different theoretical frameworks to develop it. And it specifically focuses on the topics. Right now, it's just focused on knowledge. So knowledge gained from, and it's focused on the topics that within the health education curriculum.
Yeah.
So it's
Okay.
very specific to our curriculum.
All right, no, so that sounds reasonable. And then with respect to intent to screen, are you going to develop any patient navigation system? If you're doing 3-month and 6-month follow-ups, I assume that's to find out whether or not they've been screened.
This is something that I've been still working with the community partners as far as if this is something that would be appropriate at this time to access that patient data. So far, we've been avoiding that.
No, I understand that, but yeah, because I mean, the simplest way to do it in studies that I've been involved with in the past is you just recontact the participants 3 months or 6 months later, whatever the appropriate interval is, and ask them if they had the screening.
And that's our plan so far.
Yeah, yeah. Because the issue you get into, and you know, Dr. Tufel-Schoen would be very familiar with this, is if they're part of Indian Health Service, what's the lag between when a patient calls up to make an appointment and the time until they're screened, or if it's a particular tribal health entity, you know, kind of setting it within that context.
And that's been a huge part of this, the beginning, you know, the initial development and everything. It's very collaborative, and it's based on the guidance that we've been receiving from our community partners.
Hey, well, it sounds like an interesting project. So as you write it up, I'll give you my email at the end, you know, and when you talk to Dr. Tufel-Schoen, tell her I said hello.
Will do, Denise. Thank you so much.
All right, other questions? That just seemed like a good time to address that. Okay, so let me show you in this example what we did next. It did lead us to a successfully funded R01 grant application called Epic Families, Encourage Practice and Inspire Change in Families, because now this was a family-based type 2 diabetes prevention program on excess weight, proximal indicators of type 2 diabetes risk, and lifestyle behaviors. In the proposal, we had 320 children, 160 intervention and 160 control. We did have a wait list control so that they were randomized to immediate intervention versus delayed intervention across multiple YMCA sites located in medically underserved areas in Tucson and Phoenix. So here were our 4 specific aims, and you can see how we split them out. Our primary specific aim was testing the efficacy on changes in BMI Z-score at 6 months and 12 months in the children. We examined the effects of the intervention on changes in insulin resistance compared to the controls, because that's a fundamental component of type 2 diabetes pathophysiology. We were interested in the effects of the intervention on changes in their diet quality, physical activity, sleep, and time spent in sedentary behavior. And then, as you can see, we split out evaluating the indicators of successful program implementation, including acceptability, adoption, relevance to the YMCA and participants, feasibility, fidelity, program cost, and factors influencing sustainability, because we wanted the YMCA to be able to continue this even without our grant. So as you can see, here's our participant flow and study design. We had to screen roughly 2,200 families, of which 440 were eligible because 80% were ineligible. 20 to 30% were not interested or available to participate. So then we enrolled 320 children. then we had to randomize the YMCA to the intervention or the control. Okay, because you couldn't be offering the intervention and part of the YMCA, but not another part of the YMCA. So the 20 participants were nested within a YMCA. So we had 2 in Phoenix and 2 in Tucson. So we gave them a 26-week lifestyle intervention at control. The control didn't get the intervention. Then post-intervention at 6 months, then we followed them for another 6 months. And then those that were in the control had the option to participate in the intervention. So you can see that we were randomizing cohorts, not individuals within a YMCA. So we justified the sample size based on the mean change in the BMI Z-score from the EPIC-KIT study. But now we had to adjust for the fact that we would have participants randomized within a cluster with up to 25% attrition. And our statistical testing adjusted for the 2 primary outcomes, BMI Z-score and insulin resistance, using the Bonferroni criteria. And the Bonferroni criteria is straightforward. You just take your alpha level of 0.05 and divide it by the number of comparisons that you're going to do. So to justify our sample size, in the grant application, we assumed a simplified comparison of the change from baseline to the end of intervention. Our statistical power was at least 95% for change in the BMI Z-score, assuming a correlation of the individuals within a cluster of 0.2 or below. For HOMA-IR, we needed a standardized difference of 0.9 with 90% statistical power. And we had a co-investigator who demonstrated a difference of 0.36 in obese Latino adolescents. So if we assumed a similar effect size, we would get greater than 90% statistical power. So what I've tried to do today is to kind of demystify the process of consulting with a statistician. You know, it's clear when I was talking to Melinda, we were talking about, well, you know, kind of what's the context of what you're doing, and the specific aims page, what I bring to the collaboration. You know, when you get ready to submit it, I don't know where you're submitting it. I can't help you with you know, the statistical analysis plan and statistical landmines to avoid in grant development. So my goal was to discuss statistical design considerations in the context of the proposals that you'll be developing. So I have the opportunity, let me see, well, I'm not good at this. So I have the opportunity to work with Pavel and Nicole every other week because we meet with their PI. Diana, do you want to talk about what you'll be doing and what help you might need?
Yes, thank you. So we actually have a pilot project funded by NIMHD through SHARE, through the Southwest Health Equity Research Collaborative. And Julie Baldwin is the PI of that U54 grant, NIH. And so we're working with a Native American community in Southwest, and it's also as Melinda's case, it's a small community, and we are right now in this pilot project. We are just measuring. It's actually related to Paul a little bit as well. We are doing microbial ecology in drinking water samples in this community. So we are targeting microbial community in general, just like a very broad perspective on basically who is there in the drinking water samples. So the community is around 1,000 people, but this is again pilot project, and microbial community analysis through DNA and RNA can be very expensive. So we are doing some analysis for 20 different houses plus 10 samples for like input and output from the water treatment plants. So we have like 30 samples in total for this community, and we just want to know like if there is any correlation of what we're finding in these samples with other water characterization, chemical and physical, like pH, hardness, salt, all these. So this pilot is not measuring like health directly yet, but the idea will be to go there at some point. And we're also partnering with the community to do some mitigation strategies in case we need mitigation strategies. So there is a big anthropological component, collaborative, very, very strong collaboration with the community. So we have two co-PIs are actually anthropologists that are helping me with that space. So the idea will be to go to an R1 soon. We are actually in the second year of this pilot project. We have already good results, and yeah, I would love to have like your help in like, okay, how to make this statistically valid for sure. One of the questions that I actually was having while you were presenting and while also Melinda was asking is what happens, like you were explaining about this, how we can like kind of do like a randomized selection, for example, in water. It will be like, okay, maybe we can take some randomized houses, right? But then my question was gonna be how can we account for the logistics of it, meaning that maybe some people don't want their water to be tested, so we have just to go with whoever agrees to have the water tested, and that is like not as randomized as before. So that's kind of like one big question that I have, and the second question will be like, how many well, I guess this is too big of a question, but I'm trying to see how this will work, but I'm guessing like it will be a lot of thank you, Melinda. So it will be a lot of houses that we will need to measure. So logistically, how that will look like if the community is not, I mean, they're like great collaborators, they're very open, but maybe going even beyond that we're already doing might be harder and harder while we approach like a bigger project.
Yeah, and those are really good questions that you're asking. Geographically, how large is the community?
It's not that big. I mean, my brain works in square meters, so it's a little bit different.
No, that's okay.
But I will say, well, in acres it's around 300 acres, so I guess it's not that big.
Yeah, because I'm going to answer your question in two parts. Okay, because I'm thinking about it from a statistical perspective. And then we'll talk about it from a sampling perspective. So from a statistical perspective, what I would want is I would take, I mean, I'm going to assume, I don't know if you can see me making a square with my fingers. Okay. But what I would want from a statistical perspective is to make sure that you have samples from throughout the regions. Okay. So that, you might target, if you have households, in your 300 acres, if you have households on, say, and an acre in the middle, you might want to target households in that acre, and then households, at the extremes and things like that. Especially, and I'm not an expert in water quality, but there are people who are, especially if there are geographic or geological features that you feel would impact water quality, like things like ponds or, buttes or something like that, that might impact the water quality. Then, so that's the way I would think about kind of my target. Then you ask a really good question. And this may be related to what Melinda wants to do also. You're going to need to find out who's willing to be part of your study. So you would need to be able, and it might not be feasible to include all households. So then, how many households do you have in this area?
It's around 250.
150?
200.
Yeah, okay. 250. So is it feasible for you to sample all of them?
Oh, yeah, with a big budget, I think it might be feasible, yes.
Okay, but not everyone will agree.
Exactly. Yes.
So, okay, let's say half of them will agree. That's probably about right. It's okay if they agree to have it sampled. I mean, the studies that I've seen and is, yes, you actually sample the water from all of their taps, but you also have to sample any sources of bottled water, right? That they have in their refrigerator. I mean, do they have bottled water? Do they have
They
refrigeration?
consume a lot of bottled water, so that's a great point that you're actually making there. And it's like which water they're actually consuming. So yes.
Yeah, which water are they actually consuming? Because there's the water they're consuming, there's the water they're bathing in, there's the water they're washing their dishes in. So that's why water sampling is so complicated. So then if you sample the household, what questionnaires are you asking them to fill out?
So the goal will be, yeah, like, well, they're like those two different parts. Like one is like, are there any types of pathogens that throughout the water system, meaning like from source, then treatment, then before going to the house, and when it's going into the actual tap, do we see any difference in the microbial communities in the water, and are they any pathogens in the water that they are consuming? That's kind of the biggest question.
Well, yes, that's your big question, but are you gathering any information from the participants in the household?
Okay.
Or just you're asking to sample their water?
So yeah, I was gonna go to like, so that's the first part, just like very straightforward sampling. The second part is, yes, like interviews to people that are well, to people that are in this community, how they're using their water, which type of water they're using, and how much water they're using, what are their perspectives about the water that they're using. So yeah, that's the anthropological part of it, and the questionnaire that we're using is kind of like a systematic approach of water use in communities.
Okay. So you are asking, because I'm thinking for the first part, you don't even need to use the household. You could sample it from the street. So, but then it's the second part that, yes, you're going to need to have their informed consent. So, I would say it would be appropriate for you to consider, and this is just for you to consider, identifying those who are willing to give their informed consent. And if you can accommodate all of them, then do it. There's no reason for you to randomize. But if you can't, then randomly select within an acre. I'm just using that as an example, however many you can do within each.
Okay.
Yeah.
Perfect. This is very useful. Thank you.
So Nicole and Pavel, I mean, we talked so much, but do you have any, any questions you want to ask without the boss looking over your shoulder?
I guess we do talk a lot, but I think a lot of the times it's on other projects that I'm not writing grants for. So I am trying to, I know Pavel brought up the microbiome question earlier, but I'm trying to dive into a little bit of epidemiology because I got feedback from my K99 reviews that there wasn't strong enough justification on whether there were disparities between certain racial ethnic groups for chronic pelvic pain. And so I was told that potentially rewriting a first aim with a validated questionnaire to look at overall prevalence. That said, I am not super familiar with trying to figure out the best sample size for that sort of questionnaire. I did look into, I think when I first submitted it, I looked at the overall prevalence, but as you may know, like some of these benign gynecologic conditions have very large ranges of prevalence. Like it'll be from 8% to like 30%. And so trying to decide which sort of prevalence I should be utilizing to assess my sample size. Do I do like a median or a mean of the ranges? Or, yeah, what are your thoughts?
I would use the median because I have a feeling that the prevalence is probably skewed. So a mean wouldn't work as well. So yes, I would use the median. Are you working with Leslie Farland?
Yes, she's gonna be on my mentoring.
Yeah, good. Okay, because I would talk to Leslie about this because I mean I know she's about to go away on maternity leave. But I would talk with her about it because I'm sure she has some expertise in terms of questionnaires but yeah, that sounds like a nice first specific aim. Is to just describe the prevalence of these conditions across ethnic groups.
Yeah. Okay, well, thank you.
Okay. Pavel?
I don't think so.
Okay. All right. Well, I mean, we'll talk again in a few weeks. So I think my next slide. Oh, yeah, that's how you get in touch with me. D-Roe. Oops. Well, that was my next slide. droe@arizona.edu. I am a part of NACP, so I am very willing to help. I mean, Nicole and Pavel, I'm already working with, but Diana and Melinda, I am very willing to help you. The only caveat is I am retiring at the end of June. So it's better for you to get it to me sooner rather than later. And, the Cancer Center is working, the Cancer Center and the College of Public Health are working to replace me. But especially since we've talked about it today, I'll be able to get it to it quicker than someone new.
Thank you.
Thank you.
Okay. Ron and Maria, is there anything else?
Yes. So you guys all, I don't know if you can see in the chat, Kelly has left a link for evaluation. There's Kelly. Kelly, you want to say anything about it?
We take just probably 3 minutes to fill it out. It helps give us important feedback to know how we're doing and what's working and all the things. But reach out if you have any questions, please.
Yeah, and I'll just add, I know Nicole and Pavel are well versed in this, but Diana and Melinda, any questions or you need any help connecting with Denise for any reason, we're here, guides, to support you to do that. And feel free to reach out to Ron or myself or Maria Jackson and Janie so that we can get an answer for you quickly.
Thank you so much. Appreciate that.
Very informative, Denise.
Yeah, great. Thank you, Denise.
All right, well, thank you for the opportunity, and yes, especially Diana and Melinda. You don't have you don't have a standing appointment with me on your calendars the way Nicole and Pavel do. So just reach out.
Thank you. Will do.
Thank you.